Hello everybody, welcome to my channel Omni Bio Entrance, and today we will be discussing CUCET MSc Entrance question paper with the subject code PGQP74, and it is for the subject Biochemistry conducted in the year 2022. If you are new to my channel, please do like, share and subscribe. During translation termination, after the release of RF1, RF3, that is release factor 1, release factor 3, it is found in bacteria. Is also released by hydrolyzing. RF3 is also released by hydrolyzing its GTP. Dash binds at A site and then dash bind at A site, which help in translocation and helps in the removal of E site and P site tRNA. So they have asked what happens in translation termination. So the answer here will be EFG, GTP and RRF. RRF is ribosome recycling factor. So EFG, GTP, it binds at A site, and then RRF binds at A site, which help in translocation and removal of E site and P site tRNA. Then next one, delta G0 for the reaction is 11.7, R is 8.314. The reaction is, so the delta G is negative, so naturally it is an exergonic reaction at both the temperatures 35 degree and 25 degrees. Next one, on the basis of the following hydropathy plots for three proteins A to C, predict which would be membrane protein. So this is the hydropathy index, and this is the first amino acid residue in the window. Now if you see this plot, you can see that there is some amino acids which are above the basal line, some which are below the basal line. So those above the basal line, they are hydrophobic, that is they have positive values for hydropathy index, and those below this 0, that is this basal line, they are hydrophilic or water loving amino acid, and they have negative value. Now if you take membrane proteins, there are integral membrane proteins and peripheral membrane proteins. Here if you check the graph, once I explain it, you will come to know it is for integral membrane proteins, not for peripheral membrane proteins. So integral membrane proteins are partially or completely embedded in the lipid bilayer. So the region which is embedded in the lipid bilayer, it will be hydrophobic, and the region which is above the lipid bilayer or on top, you will find that it is hydrophilic amino acids. So you should get both the amino acids for integral membrane proteins. That is, you should get a curve above the basal line and a curve below the basal line, showing it is a transmembrane protein. Whereas in peripheral protein, they are found lying on the surface. So there are no hydrophobic amino acids for peripheral proteins. So here the answer will be option 3, because you can see that here there are many hydrophobic amino acids, that is in the transmembrane region in the lipid bilayer, between the lipid bilayer, the hydrophobic region. I told you already, these are hydrophobic amino acids and these are hydrophilic amino acids, whereas here that is not the case. You can see so many hydrophobic and hydrophilic amino acids, so it is not a for the transmembrane protein. Here also same way. There is no sharp distinction between hydrophobic and hydrophilic amino acids. Some hydrophobic, then hydrophilic, then again hydrophobic, then again hydrophilic. Like that it is going on. But here there is a sharp distinction between hydrophobic and hydrophilic amino acids. So naturally third is the answer. This will be your region which is embedded in the phospholipid bilayer, and this is the region which is not embedded in the phospholipid bilayer. That is, it contains hydrophilic amino acids, and here it contains hydrophobic amino acids. So hydropathy plot for membrane proteins is option 3. Then starting with glucose labeled in either position 1, 2 or 3 with carbon 14, which labeled molecule will be the first to lose the radioactive carbon as carbon dioxide, C14 carbon dioxide, if the glucose is exclusively for the glycolytic and TCA cycles. So the glucose here, they have labeled it at one of the carbon atoms, 1, 2 and 3 with C14. Now you have to trace which carbon atom is lost from the glucose molecule as carbon dioxide. So if you remember the glycolytic pathway, you will know that there is splitting of this fructose 1,6-biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate of this glucose molecule. So here you have got this is the dihydroxyacetone phosphate and this is glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate, the carbon atoms are derived from 1, 2 and 3 of your glucose molecule, whereas for glyceraldehyde 3-phosphate, these 3 carbon atoms are derived from 4th, 5th and 6th carbon atom of your glucose molecule. Now what happens next is dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by triose phosphate isomerase. So after the triose phosphate isomerase reaction, C1, C2, C3 of the glucose, remember C1, C2, C3, that is carbon 1, carbon 2, carbon 3 of glucose molecule are chemically indistinguishable from carbon 6, carbon 5, carbon 4 respectively of the glucose molecule. That is C1 of glyceraldehyde will be derived either from the 4th or 3rd glucose molecule, carbon molecule of glucose. C2 of this glyceraldehyde will be derived from the 5th or the 2nd carbon atom of glucose molecule, and C3 of glyceraldehyde will be derived from the 6th or the 1st carbon atom of glucose molecule. Now glyceraldehyde 3-phosphate, it undergoes various reactions and give rise to pyruvate. This is pyruvate. Pyruvate, before entering the TCA cycle, it is converted to acetyl CoA and carbon dioxide is released. So this carbon atom is released as carbon dioxide. Now this carbon atom comes from this glyceraldehyde carbon atom molecule, and I told you this carbon atom of glyceraldehyde is derived from either the 4th or the 3rd carbon molecule of glucose. So in the option they have not asked regarding the 4th carbon molecule. They have told only about the till 3 carbon molecule. So that is 1, 2 and 3. It is labeled to either 1, 2 or 3 carbon with carbon 14 glucose molecule. So out of that, which glucose molecule, which carbon molecule is being labeled. So C, the answer here is C14 glucose. 3 C14 glucose. Answer is option 3. Then next one. Which of the following characteristics of proofreading are essential to maintain fidelity in DNA replication? Degrading DNA in the 5' 3' direction. No. Using methylation differences to determine parent versus daughter strand that take place in mismatch repair after DNA replication. Then always having a free 3' OH end on the strand being synthesized? No, having both nuclease and a polymerase activity on the same molecule. That is correct. Which of the following characteristics of proofreading are essential to maintain fidelity in DNA replication? So fourth option is correct. Then next one. Which one of the following correctly pairs an amino acid with a valid chemical characteristic? Glutamine contains a hydroxyl group in its side chain. No, it doesn't have a hydroxyl group, glutamine. Serine can form disulfide bonds. It is cysteine that can form disulfide bonds. Cysteine contains the smallest side chain. Glycine contains the smallest side chain. Isoleucine nearly always found buried in the center of proteins. It is hydrophobic amino acid. So it will be found in the center of the proteins away from water. So fourth is correct. Then next one. Porine, a protein from the outer membrane of bacteria such as E. coli, Rhodobacter capsulatus, represents a class of membrane proteins with a completely different type of structure. Structures of this type are built from A and contains essentially no B. Here A and B are. So A is beta sheets and B is alpha helix. It doesn't have any alpha helix, but it has got beta sheets. So answer is option 2. Then next one. Calculate the pH of a 2-liter solution containing 10 ml of 5 molar acetic acid and 10 ml of 1 molar sodium acetate. pKa of acetic acid is 4.76. So you have to use Henderson-Hasselbalch equation for this. So before that you have to calculate moles of acetic acid. So 10 ml into 5 molar divided by 2000 ml or converting 2 liters to ml, because it is 10 ml over here. So that will give you 0.025 molar. Then moles of sodium acetate, you will get it as 0.05 molar. Substitute, and then pKa of acetic acid they have already given, and you will get the answer as 4.07. So the answer which is correct here is option 2, 4.06. Then next one. Immunosuppressants such as dash prevents transplanted organs from being rejected in recipients. So immunosuppressants, they are class of medicine that either inhibit or decrease the intensity of the immune response in the body. And in organ transplant, these drugs, that is these immunosuppressant drugs, lower the body's ability to reject a transplanted organ. So they have asked here, immunosuppressants such as out of these which one prevents transplanted organs from being rejected in recipients. So it is cyclosporine. If you see the options itself, thrombin, you know it is involved in blood clotting. Aspirin, it is an analgesic and an antipyretic. So naturally cyclosporine will be the answer. So the answer will be option 2. Next one. If you have to find out whether a given protein is bound to a given gene in chromatin, which technique would you prefer using? So they have asked for a protein which will bind to DNA. So here they have asked for protein is bound to a given gene in chromatin. So the answer here will be CHIP, that is chromatin immunoprecipitation. It is a type of immunoprecipitation technique that is used to investigate or to study the interaction between DNA and protein in a cell. It can also be used to determine whether specific proteins are associated with specific genomic regions such as transcription factors on promoters or other DNA binding sites. So that is with regard to chromatin immunoprecipitation. So the answer here is CHIP, CHIP, or chromatin immunoprecipitation. Then next one. The hexose monophosphate shunt and the enterin duodraft pathway are alternative mechanism for. So they are alternative mechanism for energy metabolism in certain species of microorganisms. Answer is option 3. Which among the following is not required for prokaryotic expression vector? Not eukaryotic. They have asked for the prokaryotic expression vector. That is your bacterial expression vectors. So here the answer will be CMV promoter. CMV promoter, it is for constitutive expression of genes in mammalian cell lines. So it is eukaryotic. So that is not required for prokaryotic expression vector. Then next one. Microtubules mostly are made from. They are made from 13 protofilaments. That's a direct question. Next one. What is the key regulatory component of an operon model of regulation of gene expression in prokaryotes? So naturally it is the operator. Operator, which binds to the regulatory protein and can either repress or activate transcription. So it is the key regulatory component of an operon model. Then next one. Which of the following protein do not represent quaternary structure? Myoglobin. Myoglobin, it is tertiary structure. So the first answer, first option itself is correct. Lactate, dehydrogenase, hemoglobin, aldolase, all these have quaternary structure. Myoglobin is the one that is having tertiary structure. Then if H is enthalpy, T is temperature, the relation between entropy and Gibbs energy that you are studying from your +2 onwards. So or before that I think. So the answer here will be delta G is equal to delta H minus T delta S. Then again another question from +2. At normal temperature and pressure, 1 mole of a gas occupies a volume of 22.5 liters. Answer is option 3. Then kcat. Kcat or catalytic constant. It is defined as the number of maximum substrate molecules converted by the enzyme molecule into a product per unit time at the saturation of substrate on the enzyme. So kcat is measured under saturating substrate. Answer is option 2. Peptide bond, it is a covalent bond. Option 1. Which of the following is the storage form of a lipid? So it is triacyl glycerol. Triglycerides. So the answer here is option 3. Then next one. During mating between HFR strain and an F- strain recipient cell, F plasmid is transferred from donor to recipient cell. Chromosomal DNA is transferred from donor to recipient cell. A complete copy of F plasmid and chromosomal DNA is transferred. Both F plasmid and chromosomal DNA are transferred partially. HFR is high frequency of recombination, and when F plasmid is integrated with the chromosomal DNA in bacteria, it gives rise to the HFR bacteria. In the cross between HFR and F- cell, the frequency of transfer of the whole F factor is very low. Now why does this happen? Because the mating bridges are structurally fragile and gene transfer is incomplete. So when the mating bridges become structurally fragile, they break off very fast. So the gene transfer doesn't happen completely. So the answer here will be both F plasmid and chromosomal DNA are transferred partially. Answer will be option 4. Then next one. Identify the most simplest nutritional category of microorganisms. So it is photoautotrophs. They are the simplest nutritional category of microorganism. The recommended percentage of dietary fat per day is, so it is 20%. Then next one, the counterparts of DNA polymerase 3 in eukaryote is or are. So they are polymerase delta and epsilon. Answer is option 2. Why do erythrocytes swell and burst when placed in water? Since solute concentration is lower outside, by passive diffusion water moves inwards. That itself is the right answer. Serum albumins and globulins can be separated by the method of electrophoresis. So serum protein electrophoresis, it is an inexpensive method for separating protein based on their net charge, size and shape. So the two major proteins that are present in the serum are albumin and globulin. Albumin is the major component and it lies closest to the positive electrode. And globulins, there are 5 globulins, that is alpha 1, alpha 2, beta 1, beta 2 and gamma. And of these, the gamma fraction globulin, it is found closest to the negative electrode. So you can separate albumin and globulin by electrophoresis. Next one, SH2 domains specifically bind to. So SH2 domains, they are predominantly involved in tyrosine kinase signaling pathways and they recognize phosphorylated tyrosine residues within their targeted proteins. So SH2 domains specifically bind to phosphorylated tyrosine residues. Then next one, which of the following is the component of nucleoprotein? So nucleoprotein, they are conjugated protein and consist of proteins linked to nucleic acids. That is either DNA or RNA, and the protein which is combined with DNA is nothing but your histones or protamines. So which of the following protein is a component of nucleoprotein? It is your histones. Answer is option 3. Which ratio is constant for DNA? A plus G by A T plus C. Chargaff's ratio. So answer is option 1. Then next one. Which of the following molecules bind strongly to avidin? So avidin, that is streptavidin. You might remember it. Biotinylated. Okay, all these terms if you remember, it is biotin, that is bind strongly to avidin. Then next one, Tay-Sachs disease is the result of genetic defect in the metabolism of. So Tay-Sachs disease, it is a neurodegenerative disorder and it is because of the enzyme deficiency of the enzyme hexosaminidase A. And because of this deficiency of this particular enzyme, it results in the accumulation of GM2 ganglioside. Now this hexosaminidase, what it does is actually in normal condition, it is responsible for degradation of GM2 ganglioside. So Tay-Sachs disease is the result of genetic defect in the metabolism of ganglioside. So answer is option 2. Then which vitamin is derived from cholesterol? It is vitamin D, that is derived from cholesterol. So answer is option 3. Then next one, starch and glycogen are both polymers of alpha D glucose. Ascorbic acid deficiency is associated with, ascorbic acid deficiency is vitamin C deficiency and it is not associated with bone deformities. They have asked for it not associated. So it is not associated with bone deformities. Then next one, which of the following coenzymes are not associated with pyruvate dehydrogenase complex? So there are 5 different coenzymes. They include thymine pyrophosphate, lipoic acid, coenzyme A, FAD and NAD. So out of these, biotin is the one which is not associated with pyruvate dehydrogenase complex. So the answer here is option 4. Then next one, Which of the following enzymes are not regulated by phosphorylation, dephosphorylation? So out of this, hexokinase. It is not regulated by phosphorylation, dephosphorylation mechanism. Glycogen phosphorylase, pyruvate kinase, glycogen synthase, all these are regulated by phosphorylation, dephosphorylation mechanism. Then next one, inheritance from father to son is not possible in which of the following genetic conditions? So phenylketonuria, color blindness, Down syndrome, Huntington's chorea. All of these are autosomally transmitted, whereas color blindness is an X-linked inheritance, and in X-linked inheritance there is no male to male transmission. So from father to son is not possible. So the answer here is option 2, color blindness. Then next one, which of the following hormones will not mediate their effect via G protein linked receptor pathway? So now it is thyroxine, which is not having any G protein linked receptor pathway. Then next one, tyrosine is not a precursor for which of the following molecules? So melatonin. Tyrosine is not a precursor for melatonin. Answer is option 1. Then which of the following is not true about conjugated bilirubin? Deficiency in this conjugated bilirubin result in Crigler-Najjar syndrome. That is correct. More soluble in aqueous solution than unconjugated form. That is also correct. And the conjugated bilirubin is elevated in obstructive jaundice. That is also correct. Transported in blood bound to serum albumin. So that is the incorrect statement. Answer is option 4. Then next one. Which of the following is not a metalloenzyme? Now xanthine oxidase, it has molybdenum and iron. Lysyl hydroxylase, it has iron only. Alcohol dehydrogenase, it has zinc. But lactate dehydrogenase doesn't have any metals in the enzyme. So the answer here is option 4, that is lactate dehydrogenase. Then next one. Formation of mature insulin includes all of the following except removal of signal peptide. Yes. Alpha carboxylation of glutamate residue. No. Disulfide bond formation, yes, and cleavage of peptide, that is also is involved. So the answer here is option 2. Then next one, assertion reason type questions. Assertion A. ATP binding is essential for the muscle relaxation event in the muscle function. That is correct. The process is driven by cross bridges which extend from the myosin filament and cyclically interact with actin filaments as ATP is hydrolyzed. That statement itself is wrong. So A is correct but R is not correct. Then next, assertion and reason. Assertion. Transcription and translation in prokaryotes is synchronous. Of course. Both are coupled processes occurring in the cytoplasm. Yes. A and R are correct, and R is the correct explanation of A. Because it is occurring in the cytoplasm, transcription and translation are synchronous in prokaryotes. Then next one. The salting out technique is one of the most commonly used methods for protein purification from a solution. Yes, that is correct. At low salt concentration, protein solubility usually decreases, leading to precipitation. It is not low salt concentration. At high salt concentration, protein solubility usually decreases and leads to precipitation. So A is correct, whereas R is not correct. The next assertion. The DNA molecule is antiparallel and a double helical structure. Yes, that is correct. The backbone outside the double helix is made up of phosphate and sugar. Yes. So both A and R are correct, and R is the correct explanation of A. Then next one. IgG antibodies provide the passive immunity and protect the fetus. It crosses the placenta. Because it crosses placenta, it provides passive immunity and protection to the fetus. So both A and R are correct, and R is the correct explanation of A. Then next one. Protein synthesis is a central function in cellular physiology and is the primary target of many naturally occurring antibiotics. Correct. Tetracyclines are class of broad-spectrum antibiotics used in the management and treatment of a variety of infectious diseases by inhibiting translational machinery in bacteria. So that is also correct, and it is the correct explanation of A. So both A and R are correct, and R is the correct explanation of A. Then next one. Vesicular transport is the predominant mechanism for exchange of protein and lipid between membrane-bound organelles in eukaryotic cells. Okay. Golgi-derived vesicles are involved in several vesicular transport steps, including bidirectional transport within the Golgi and recycling to the endoplasmic reticulum. A protein X is the type 2 transmembrane protein localized to the Golgi. If you add KDEL sequence to its N-terminus, the mutant protein will localize in the Golgi. They have told that if you add a KDL sequence to its N-terminus, the mutant protein, the protein which is mutated, it will be found in the Golgi, else it will be transported to the endoplasmic reticulum. Now the reason. Endoplasmic reticulum luminal protein, especially those present at high concentration, can be passively incorporated into COP2 vesicles and transported to the nucleus. Many such proteins bear a C-terminal KDEL sequence that allows them to be retrieved. The KDL receptor, located mainly in the nuclear network, binds proteins bearing the KDL sorting signal and returns them to the endoplasmic reticulum. Here, the KDL receptor located mainly in the nuclear network. It is not the nuclear network KDL receptor is present. It is present in cis Golgi, lysosome and secretory vesicles. So this statement is wrong. But A is correct. So A is correct and R is not correct. So the answer here is option 3. This topic is taken from transport of proteins. Then next one. What is the correct set of enzymes involved in base excision repair? So DNA glycosylase, AP endonuclease, DNA polymerase 1 and DNA ligase 1. That itself is the answer. The genome of viruses, prokaryotes and eukaryotes could be circular linear double-stranded RNA and linear double-stranded DNA. RNA is not present in eukaryotes as a genome. Then, so first option is not correct. Then linear single-stranded DNA, double-stranded DNA. Single-stranded DNA is not present in eukaryotes as a genome. So that also is not correct. Circular linear, again single-stranded DNA. So that is not correct. Linear circular double-stranded. That is correct. Linear can be present in viruses. Prokaryotes it is usually circular and eukaryotes it is double-stranded DNA. So the answer here is option 4. Then given below are two statements. Which statement is correct, or if both the statements are correct, then that. So the first statement, the alpha helix, beta strand and sheet and beta turn are the most prevalent elements of protein secondary structure. Yes. Secondary structures are stabilized by hydrogen bonds between atoms of the peptide backbone. That is also correct. Alpha helix and beta sheets, they are secondary structures and they are stabilized by hydrogen bonds. Fine. Protein tertiary structure results from hydrophobic interactions between non-polar side groups and from hydrogen bonds and ionic interactions involving polar side groups and the polypeptide backbone. These interactions stabilize the folding of the protein, including its secondary structural elements, into an overall three-dimensional arrangement. That is also correct. So both statements 1 and 2 are correct. Then next one. Monoclonal antibodies which bind one epitope of an antigen can be secreted by cultured cells called hybridomas. Of course. So that statement is correct. These hybrid cells are made by fusing helper T lymphocytes. It is not T lymphocytes, it is B lymphocytes with immortal myeloma cells and then identifying those clones that produce the desired antibody. So statement 2 is incorrect. So statement 1 is correct and statement 2 is incorrect and your answer is option 3. Then next one. Statement 1. Immunofluorescence microscopy make use of antibodies to localize specific components in fixed and permeabilized cells. That is correct. Direct immunofluorescence microscopy uses an unlabeled secondary antibody followed by fluorescently labeled primary antibody that recognizes the secondary one and allow it to be localized. In direct immunofluorescence, the primary antibody is directly coupled to the fluorophore, allowing for easy handling and quick visualization. No secondary antibody is involved in direct immunofluorescence microscopy. So the statement 1, that is wrong. We will see statement 2. Fluorescence resonance energy transfer is a technique in which light energy is transferred from one fluorescent protein to another when the protein are very close, thereby revealing when two molecules are close in the cell. That is correct. So statement 1 is incorrect whereas statement 2 is correct. So answer here is option 4. Then next one. Nucleoid is not present in animal cells. Who told? Nucleoid is present in mitochondria. So animal cells are organized into clusters of 5 to 7 genomes referred to as nucleoids in mitochondria. So first statement is incorrect. The nucleoid is a chromatin-dense area within the cytoplasm and contains the bacterial DNA. That is correct. So statement 1 is incorrect and statement 2 is correct. Next one. Statement 1. cGMP is involved in the perception of light. Yes. Cyclic GMP gated channels in the outer segment membrane are responsible for the light included changes in the electrical activity of photoreceptors. So both statements 1 and 2 are correct. Then next one. Among the two nucleic acids DNA and RNA found in living cells, DNA is considerably more stable than RNA. Yes. Double-stranded state of DNA and single-stranded state of RNA contribute to this status. Yes. So both statement 1 and 2 are correct. Polymerase chain reaction and real-time polymerase chain reaction are techniques. Of course. So that is correct. They have invented for in vitro amplification of DNA as well as RNA. For RT-PCR, it is for rapid sensitive determination and quantification of nucleic acids. So it is not involved in any amplification of DNA and RNA, whereas PCR is involved in amplification of DNA. So statement 1 is correct, whereas statement 2 is incorrect. Diethyl aminoethyl cellulose is typically used in ion exchange chromatography for the separation and purification of proteins. Yes. DEAE cellulose is cationic exchanger. It is an anionic exchanger, not a cationic exchanger. So statement 1 is correct, but statement 2 is incorrect. So option 3 is the answer. Next one. The Gibbs energy change of a process can be determined by performing measurements under equilibrium conditions. Yes. Gibbs energy change at equilibrium is 0. So both statements 1 and 2 are correct. Then next one. In non-competitive inhibitor, Km remains unaltered while Vmax is reduced. That is correct. In statement 2, Km increases, Vmax remain unchanged. That is wrong. So statement 1 is correct, but statement 2 is incorrect. Option 3. IgM antibody is pentamer. It has 5 combining sites. It has 10 antigen binding sites, that is combining sites. It is a pentamer. So each antibody it will have 2 combining sites. So it is a pentamer. It has 10 antigen binding sites. So statement 1 is correct, but statement 2 is incorrect. So answer is option 3. Then next one. Free living cells usually incorporate free DNA molecule in its genome. This process is known as transformation. Both statement 1 and 2 are correct. Answer is option 1. Then next one. They have given chromatography and dialysis. You have to see how they take place, how they are the principle behind their functioning. So ion exchange chromatography, it is net surface charge. Based on the net surface charge of your protein molecule, you can use either cationic or anionic exchange chromatography. Dialysis, removing of waste products, of course. Affinity chromatography, specific binding interaction between an immobilized ligand. So it is a ligand receptor method of binding is involved in affinity chromatography, and thin layer chromatography is based on the principle of differential partitioning. So the answer is option 1. A2, B4, C1, D3. Then next one. Of these enzymes, which is hydrolase, which is oxidoreductase, transferase and ligase. So hydrolase is lipase. Lipase is a hydrolase enzyme, A4. Oxidoreductase is cytochrome oxidase. Transferase is pyruvate kinase, and ligase is acetyl-CoA synthetase. So the answer here is option 4. Then what happens during the various phases, growth phases, various phase of growth? So lag phase it is adaptation to growth condition, A3. Log phase, it is exponential phase, it is also called exponential phase. Stationary phase is where you have depletion of essential nutrients, and death phase is the decline phase. So the answer here is option 1. A3, B1, C2, D4. Then here they have given various organisms and the diseases that they cause. So Mycobacterium, it causes tuberculosis and leprosy. Mycobacterium tuberculosis causes tuberculosis, and Mycobacterium leprae, it causes leprosy. So A is 2. Retrovirus is involved in causing AIDS. Plasmodium is for malarial fever, and Vibrio cholerae is diarrheal illness. So the answer here is option 3. A2, B1, C4, D3. Okay, various enzymes involved in these cycles. So in glycolysis, which enzyme is involved? Out of the given one, it is pyruvate kinase, A2. In TCA cycle, it is isocitrate dehydrogenase. Urea cycle, it is arginosuccinate synthetase. And Calvin cycle, it is phosphoribullokinase. So the answer here will be A4, 4, that is D2, B1, C4 and D3. Then coming to various cells. B cells. B cells is found in bursa of fabricus in birds. T cells, they mature in the thymus, B4, A3. NK cells, natural killer cells. They are tumor cells, found in tumor cells, and monocytes, they are tissue macrophages. So the answer here is option 1. Okay, then various types of transporters. ABC transporter. ABC transporter is cystic fibrosis. A defect in ABC transporter result in cystic fibrosis. A3. P-type ATPase. P-type ATPase is Na plus K plus ATPase. F-type ATPase is ATP synthase, and passive transporter is a glucose transporter. So the answer here will be option 3, that is A3, B1, C4 and D2. Then polymerase and their properties. DNA polymerase 1. DNA polymerase 1 is 5' 3' exonuclease activity. DNA polymerase 2 is least molecular weight 80,000 Daltons. And DNA polymerase 3, it is highest polymerization rate 250 to 1000 nucleotides per second. And DNA polymerase 4, DNA repair translation synthesis. So the answer here is A2, B3, C1 and D4. Now cyclic AMP. Cyclic AMP is forskolene. Calcium is calmodulin. B4. 1,2-diazyl glycerol is forbol ester, and 3', 5' cyclic GMP is nitroglycerin. So the answer here is option 4. A3, B4, C2, D1. Then ions and their concentration. So K plus, it is at concentration of 145 to 155. Na plus at 10 to 12. Cl minus is 3.8 to 4. And Ca2 plus is 0.001 to 0.003. So the answer here is A1, B4, C3 and D2. You have to match RNA polymerase, which are will result in their synthesis product. So RNA polymerase 1, it is pre-rRNA. A2. RNA polymerase 2 is mRNA, siRNA, miRNA. RNA polymerase 3 is tRNA and snRNA, and DNA polymerase gamma. It is for mitochondrial DNA replication and repair. So the answer here is option 3, A2, B1, C3, D4. Okay, G alpha S. G alpha class and its receptors. G alpha S, beta adrenergic epinephrine receptor for glucagon, serotonin, vasopressin. Then G alpha I, alpha 2 adrenergic receptor, muscarinic acetylcholine receptor. G alpha Q, it is alpha 1 adrenergic receptor. G alpha tau, it is rhodopsin in rod cells. So answer here is A2, B3, C4, D1, option 4. So that's it. Thank you for watching my channel. Please do like, share and subscribe. I have also uploaded life science, then plant biotechnology and applied microbiology also. You can go through them. Okay, thank you. Thank you for watching my channel.